And so we even went into taking biopsies of their airway epithelia, their nose epithelial cells, and ex vivo infected them with SARS-CoV-2. And indeed, the virus cannot replicate. With the advent of high throughput sequencing, we've essentially been able to first sequence our entire exome, which is the part of our genome which is coding. I like to tell the story about this form of cancer, which was autosomal dominant, so inherited from one generation to the other. And it was very unique in the sense that it was a self-healing cancer.
Hello, and welcome back to the MGI Tech Podcast. This episode for Rare Disease Day, we're talking to Bruno Reversade from KAUST in Saudi Arabia, whose work focuses on studying rare Mendelian genetic disorders to give us a better understanding of more common conditions, which can lead to some really interesting breakthroughs. To start off with, could you just give us a bit of an introduction to yourself and the work that you do?
My name is Bruno Reversade. I am now based in Saudi Arabia in KAUST, and I have been working for the last maybe 15 to 20 years in the field of human genetics, particularly focusing on Mendelian disorders, meaning diseases or traits which appear to be caused by a single gene defect.
So, a big theme in your research is the concept of gaining insights into common diseases by studying rare diseases. Can you tell us a bit about how that comes about, how that process works and how you find widely applicable insights from studying rare disorders?
Yeah, so Mendelian disorders or single gene knockouts or gain of function are rare, and it takes a lot of effort to understand how their study can actually illuminate more common and more complex diseases. But it's a very strong stepping stone into understanding the biology that underpins a lot of complex diseases or large unmet medical needs. I could probably illustrate this paradigm, which I call rare begets common, by showing a series of illustration so that people can understand, perhaps. One of the first one that we began with was the study of a very rare and unusual form of progeria or progeroid condition. So a condition in which the children are born with a aged appearance and seem to age excessively. Aging is a very complex phenotype, right? And it affects many different lineages, different organs in different ways. And so while this is extremely rare condition, it was actually known as cutis laxa, so a disease where the skin appears to be excessively wrinkled. We were able to identify several families across the world, mainly in the Middle East, because of a large degree of consanguinity. And then we identified the causative gene, which turned out to be an enzyme involved in proline metabolism, so how the cells metabolize proline. But through many years of work, we realized that the same gene or the same protein encoded by this gene was very commonly hijacked in the context of cancer. So here we are with an extremely rare condition causing premature aging, where the gene is lost. But the same gene is actually leveraged upon by cancer cells to fuel tumorigenesis. So if you put the two together and then you say, well, why wouldn't I apply the concept of premature aging to the tumors themselves? Since the tumors are relying on the same pathway to fuel their growth, what if I remove this gene, as I have discovered in patients that are aging prematurely, will I be able to essentially blunt tumorigenesis? So by studying an extremely rare premature aging disease, we have validated a novel drug target for a common indication, which is cancer in solid tumors. So knowing now that the loss of this gene might actually blunt tumorigenesis, we have gone into a drug development campaign to block that protein in the context of common forms of oncology. So I've gone from an extremely rare condition to a potential therapeutic intervention for a large unmet medical need, which is solid tumors. And in this instance, it was particular for hepatocellular carcinoma, which is the most common liver cancer.
Mm-hmm.
I hope it was clear how
Yeah,
the
yeah,
journey from a rare to a common.
yeah, definitely. And that example was really helpful, just to sort of see how that works.
Yeah.
So, could you explain to us the difference between Mendelian diseases, which have this sort of fairly direct causality between a change in a gene and the disorder, and quantitative trait loci studies, which are more associative, maybe it's less direct genetic link?
Yeah. So in the field of human genetics, you can approach a trait or a phenotype from the angle of single gene effect, which are fully penetrant and therefore causative. Or you can go about it looking at association, meaning finding alleles or variants in the genomes of common sporadic forms of these phenotypes, and determine how correlated they are with the appearance or appearance or progression of that phenotype. It's a bit complex what I'm explaining, but essentially you can decide to look for the fully penetrant and single gene effect, or you can go through an approach which is called GWAS, genome association studies, where you will find common variants, but with very small effect size. So the variants that you will find are going to be proxies for regions in our genome that may have an influence on a trait, but they might not be causal. And so there is a divide between GWAS and mechanistic studies, because it's very difficult to go from a GWAS hit to understanding how this variant might actually be influencing the phenotype of choice. Whereas in Mendelian or monogenic diseases, you know that the single variant is actually the cause of the phenotype that you study. So you have a much more direct genotype-phenotype correlation.
So, uh, how do you use gene testing methods and gene sequencing? In your studies, how do those sort of technologies, those methods help with the work that you do?
Well, it's a paradigm shift. For the longest times, and this is before my time, people were limited by the fact that they could not readily sequence large portions of the genomes. So they would essentially be able to narrow down a trait or a phenotype to a particular chromosome, and that would be the end of it. And in the last 15 to 20 years, with the advent of high throughput sequencing, we've essentially been able to, within a matter of days or hours, essentially first sequence our entire exome, which is the part of our genome which is coding, which has really introduced a paradigm shift in the efficacy or in the amount of diagnostics that we can reach. And now we're going for whole genome sequencing. So we're not only limiting ourselves to 2 to 3% of our genome, but the entirety of what constitutes our blueprint, DNA blueprint. And now we're going even beyond that, because we are able to not only sequence our genome, We can, in parallel or even in a synchronous manner, you can actually sequence your transcriptome. You can sequence your epigenome, your methylome. So you have a wide array of possibilities. And when you triangulate all these data, you have a very profound, granular understanding of how our genome behaves, not only from the perspective of the sequence, but in a lineage dependent manner. How is this gene expressed in the liver as opposed to the central nervous system, as opposed to the skin? So we're very fortunate to be able to question this in different organs.
Yeah. So, speaking of sort of paradigm shifts, uh, with this, availability of sequencing technology, along with the approach you talked about, where you can get sort of ideas for new drug targets by studying Mendelian diseases. Um, what implications do you think this is going to have for the future of drug development over the coming, maybe decade? How is it going to change the landscape uh, in terms of being able to validate drug targets um, through sequencing and through studying single gene disorders? Is that going to have a big change in how the drug development pipeline works? Is it going to lower attrition rates? Are we going to get more personalized drugs? How is that going to change things?
Right. Okay. So I don't know that I hold that I know what will happen, but for a fact, targets that have been validated through human genetics have a much lower attrition rate. Um, and it goes back to the concept that we have saved or cured so many mice. But those studies which have been using animal models too often, do not translate into human. So we need to pivot towards essentially human as the model of choice, and really leverage the power of high throughput sequencing and leverage the knowledge that we have from Mendelian disorders to determine if a gene is really causal for the phenotypes that we study. So the important message here is that if you start with human genetics, you're much more likely to know that the phenotype you study is really translatable for medicine, for patients. And yeah, well, that's about it.
Hmm. Yeah, I think it's something we've talked about in a previous episode. Is with personalized medicine in particular for cancer, it can be difficult when it comes to the later stages of trials for drugs uh, to validate the performance, because it's impossible to have a cohort of for the study that you're going to test against, when the whole point is you're trying to treat essentially each tumor as individual. Finding identical phenotypes across cancer patients is impossible. So does it play into that as
Mm-hmm.
well? If you can isolate the effects of individual genes by studying them, in Mendelian disorders, does that play into that as well, and sort of give you another route to validation?
um, that is probably a little bit outside of my area of expertise. um, I have not really delved into complex forms of cancers. I have looked at inherited forms of cancers where we have a single gene effect. Um, but I again, like, I can here highlight or emphasize the fact that we have looked at very rare forms of cancers, and there I like to tell the story about this form of cancer, which was autosomal dominant, so inherited from one generation to the other. And it was very unique in the sense that it was a self-healing cancer. So from one generation to the other, we would have males and females that would develop squamous cell carcinoma or skin cancers, but they didn't need any intervention. And the tumors would self-heal on their own in a matter of a couple of months. And so we said, well, that is very powerful because we have a fully penetrant Mendelian disorder, which predisposes to skin cancer, but a very particular one, which also self-heals. And so from that, we identified the gene, and it turned out to be a founding member of the inflammasome, which is a component of our innate immune system. And it's called NLRP1, that's the name of the gene. But because it is responsible for essentially telling the body that there's been an insult and therefore start inflammation, we may be in a position to leverage that knowledge and essentially at will turn on an RP1 in more sporadic forms of cancers and label the tumor as hot, meaning that it should be cleared by our adaptive immune system. So again, starting from a very peculiar and unique condition, how can we, through functional genetics and through assay development and understanding the basis of the pathway, use it for much more common indications, if that makes sense.
Mm-hmm. Yeah, that's great. So this all plays into the way you've described your approach before to studying these genetic disorders, which is kind of talking about these very rare phenotypes as nature's experiment. Can you talk a
Yeah.
little bit about that approach? What you sort of mean by that? How that applies to everything we talked about.
So Mendelian genetics, or essentially rare or extreme phenotypes, are events that happen in the general population. And so they are essentially nature's experiment. They are extreme phenotypes, and they give us the boundaries of what is possible within nature. And I like to say that they're a window into nature. They give us the boundaries of what is possible. If you think of any trait as a bell curve, we are really following the extremes of that distribution. And if you take a very simple example, if you take hair growth, which is a very common phenotype, or the loss of hair or balding, it's very complex. There are many factors that influence its outcome in males and females. But the point is that if you start with the extremes of these phenotypes, and there are two very profound examples. One is the werewolf syndrome. I don't know if you've ever seen. There's only a few families in the world, which essentially where individuals are covered with hair, their entire body, their face, and there's a gene for it. And conversely, you have alopecia universalis, which is a condition where you have lost all ectodermal derivatives. So you have no hair, no eyelashes, no eyebrows. Sometimes you have no teeth. And so these are the anchor points. And they're very profound because they're a single gene defect, and they give you a stepping stone into what might be the pathways that are affected in the common forms of hair loss, which are affecting the general population. And they give you a pathway. They give you a frame under which you can actually perhaps identify proxies and strong drug targets.
Yeah, that's fascinating. It'd be great to get just a couple more stories. Just it's really interesting to hear how these really specific genetic defects can lead to understandings about obviously in that case it's fairly clear where the link is, but other times it's not so clear how how the insight comes about. So how about the study you did on RSPO2 and how that links through to colorectal cancer? Could you tell us a bit about that one?
Yeah. So this is a story that is very dear to me. And that stems from my training using looking at early birth defects. So anomalies that begin during development, so in utero. And for that, we chose to focus on an extreme phenotype, which is called tetraamelia, which means the absence of all limbs. So we're talking about children that are born without arms and without legs. It's not a lethal. It's not a disease that actually is incompatible with life, because you can actually live without arms and legs. But it's a very profound phenotype. And so we decided to address this, and found out that it was caused by a mutation in a gene that is encoding for a well-known growth factor that affects the Wnt pathway. And through deep mechanistic studies, we found out that this gene, RSPO2, RSPonding2, was responsible for the development of limbs across evolution. So not only in humans, but also in mice, also in frogs, and so on. And through a lot of work, we also identified that what was believed to be its cell surface receptor, because it is a secreted molecule, wasn't what was believed to be. Actually, we found that there was another class of receptors that RSponding was using to direct the formation of the limbs. And the question that you should be asking, that we ask ourselves, is how does this knowledge apply to more common indication? And in this case, it was very clear, because RSpondin is the number one gain of function mutations in RSponding 2 and 3 is the most common cause of colorectal cancer in human. We found the loss of function of RSponding to be causing loss of limb development. But when you overexpress it, in 10 to 15% cases of all colorectal cancer patients, you have too much RSponding 2 and RSponding 3 signaling. And so the pathway that we dissected in the context of arm formation, arm development, and leg development is directly relevant to designing perhaps better drugs for preventing the growth of colorectal cancer. And the reason behind that is that we have a limited toolkits of pathways in our genome, and they're repeatedly used for different processes. So we use RSponding to make limbs, but we also limit its activity in order not to have certain forms of cancer. So this is why using or studying a very rare phenotype, such as limb development, can have profound implications for a common medical need, which is cancer.
Yeah, that's fascinating. And yeah, that makes sense. The kind of link with it being a growth factor, either deficient or sort of overproduced, is it's really interesting. On the surface, you wouldn't think those things were linked, but from the looking at the disorders, this is the same thing that causes cancer, just the other way around.
Yeah.
And
we had
study.
another example.
Yeah.
Another example which is telling is that we were studying children that were born without a nose, and it's called arrhythmia. And I think if you just look it up on Google, you'll see that this is a textbook phenotype which had escaped essentially genetic diagnosis for decades. And so we're talking about children that are born without a nose and without olfactory bulbs. Very profound and spectacular phenotype. We gathered many cases from around the world. We identified a single gene, and this gene turned out to be one of the cause of one of the most common form of muscular dystrophy. So again, you're facing a paradigm where the same gene is responsible for nose development, but it is also utilized later in life to prevent muscular dystrophy. And one is a loss of function for the nose, and the other one is a gain of function. So there again, you can say, oh, how can I use the knowledge of these children that have not formed a nose to perhaps boost the expression of that gene in those common forms of muscular dystrophy? So the biology is the same, but with different outcomes.
Yeah, that's fascinating. I guess again talking about like the overall approach here and how more readily available and sort of higher throughput sequencing technologies can help as well. Do you think uh, this kind of process where you're drawing these links between um, different conditions, different diseases, where the only link is this particular gene, as the sort of literature builds up and the understanding of more of these diseases increases, and more the common, maybe more complex diseases as well, we understand those better genetically. Do you think this process is going to get easier? Your sort of your treasure hunt for like, oh, this is the link. We found a gene that links them together. Do you think that's going to get sort of more productive over time, or is it going to continue to
Um,
be a bit of a a bit of potluck?
I feel that there will be no shortcuts. The tools available to us are more and more powerful, and the introduction of AI solutions might help us, but there will be no ways of. I don't think one can infer and understand a pathway without deep mechanistic studies, and that takes time and a lot of investment and a lot of manpower and brain power. So making the connection from rare to common will not will not always be immediate, and it will require some investment, significant investment, and sometimes we ourselves cannot even make it. I've seen some of our discoveries being translated by others because they had knowledge that we didn't have, and
Mhm.
and therefore, um, yes, I'm not sure that everything will be easier or more facile in the future. You still have to do a lot of work.
Yeah, yeah. It does make sense that a sort of a third party, someone like less involved, might have insights that you don't, um, when you've been in the depths of a study, um, with these sort of slightly more obscure links. There was one other example that I wanted to talk about, your work on FOCAD gene deficiency, um, that links liver disease and sort of airway infections. Could you talk a little bit about that study and what you found there?
Mm-hmm. So this is a recent publication where, again, we thought that looking at Mendelian forms of liver cirrhosis would be a powerful way to understand what are the genetic underpinnings of such a common disease. So liver fibrosis, liver cirrhosis is mostly driven by insults, such as viral infections, or diet. But this must have um, it must have a genetic underpinning. It must be working on pathways in the liver. So we decide to look for pediatric forms of liver cirrhosis. And indeed, we were able to identify a Mendelian form of congenital liver cirrhosis, meaning that there are genes which are responsible in all of us to prevent the formation of the deterioration of liver homeostasis. And one particular gene is known as FOCAD, which we in this study found was operating in a particular pathway which is known as the ski mRNA surveillance pathway. Anyway, these are details. But as we were looking at understanding how this pathway affects liver health, a paper was published in PNAS by a group in the US that showed that in cell culture, so in a very remote, heterogeneous assay, the very same pathway seemed to be important for viral replication. So they were using cultured cells and inhibiting the pathway, the same genes that we had been studying in humans, and found that, for instance, they would no longer be able to replicate viruses such as SARS-CoV-2 and other ones as well. So this piece of data got us thinking, and we said, well, if that's the case, while our patients are suffering from a very severe form of liver fibrosis or cirrhosis, could it be that at the same time they are resistant to viral replication and viral infections? And so we went back to these patients and asked their parents and actually these young children whether they had had any form of COVID or any other forms of common airway infections, such as influenza. And it turned out that they had none or very, very mild forms of them. And so while they had a very severe liver phenotype, it appeared to be that they were a natural form of resistance to these common airway infections. And so we even went into taking biopsies of their airway epithelia, their nose epithelial cells, and ex vivo infected them with SARS-CoV-2. And indeed, the virus cannot replicate. So what I'm saying here is that we studied a liver phenotype. Through literature search, understanding of what has been done in this pathway, we found out, oh, maybe it's involved in viral replication. We validated it in human, and now we have a pathway which seems to be essential in the host, that is ourselves, humans, to allow viral replication. So essentially, the virus hijacks this pathway to replicate in our airway. What does it mean? It means that perhaps we could design a drug against this target, not in the liver because it would cause liver disease, right? But if we only target it to the airway epithelia, maybe then and only then will we have a way of preventing viral replication.
Yeah, that's a really interesting one. There must be like some really great sort of eureka moments, when you're trolling through, when you're searching through the literature and you find a link like that you wouldn't know unless you went looking for it.
Yeah, exactly. And this is why you have to do due diligence, and you're essentially relying on other people's work and publications to make the link between rare to common.
Mm-hmm. That's been fantastic. Are there any other sort of stories that come to mind that you think would be good to share? Any particular cases that we haven't talked about yet that would be good examples?
Maybe an important concept is that we are very much relying on extreme phenotypes that are easily captured, or how would I say? We have a tendency to look at disease phenotypes, meaning that you have the opportunity of working with patients that no, you're consulted, or the patients go to the clinic because they have a problem, right? Because there's an issue and they would like it to be resolved. But bear in mind that at the other end of the spectrum of this bell curve, there are and there will be positive traits, which are also going to be monogenic, single gene. So there will be individuals, which I would like to call superhumans, or people that have beneficial phenotypes, and they don't go to the clinic because they don't need to, they don't complain. And these are extremely powerful as well, but they're more difficult to capture because you essentially have to look for them, and they don't actually go to the clinic, as I said. So I'll give you examples. We have seen families in the Middle East where it's an autosomal dominant trait where their circadian rhythm, the sleep to wake rhythm, are completely altered, and they only need to sleep a couple of hours at night, and they're perfectly functional. And it seems to be monogenic. So here you are. You might have a gene which is I don't know if it's a gain of function or a loss of function, but is controlling how much time you can actually function during 24 hours. Whereas most of us need 8 to 10 hours of sleep. And that applies to many, many different phenotypes. There are people that are transmitting leanness. They're always lean, right? They're not predisposed to gaining weight. And so these are also profound examples, and maybe you would find perfect novel drug targets for treating obesity by studying these extreme cases of leanness.
Yeah, that's a really interesting approach. It'd be interesting to sort of look at, how you could try and find more of those the cases. Maybe sort of just put a call out on social media, like, who got any unusual um,
Exactly. And that's the good
unusual
idea is like,
traits.
yeah. If you think that you are unique or exceptional in any particular processes, it could be it could be wound healing, it could be sleep, it could be pain. There are cases of people that do not feel pain, and they identify
Mhm.
novel drug targets for this.
Yeah, that's fascinating.
Yeah.
Great. Okay, thanks so much, Bruno. That's been a really interesting conversation. Thanks for your time.
You're most welcome. Thank you for having me.